From the Cosgun Lab and the Bioengineering Media Lab at Georgia Tech, welcome to Lab Coat Conversations, where you too can join the discussion on the next academic breakthroughs. Join your host, Hector Torres, as we talk to Georgia Tech researchers. This episode, please welcome Cheng Zhu. So we are here with Dr. Zhu, who runs the Cellular and Molecular Biomechanics Lab. The goal of Dr. Zhu's lab's research is to gain a fundamental understanding of important biological processes at the level of cells and molecules as related to human health and disease, and use such understanding to guide the development of strategies for treating disease. Dr. Zhu has a wide range of interests from the immune system, cardiovascular system, all the way to the nervous system, with a unifying focus on the mechanotransduction of cell surface receptors. His lab develops and employs a battery of highly sophisticated and relatively unique biomechanical technologies and experimental methods, including single molecule force spectroscopy and molecular dynamic solutions. Did I get most of that correct?
Yeah.
Great. So for the first question, can you define for our listeners maybe a little bit about mechanobiology and how it pertains to you and your lab?
Yeah, so mechanobiology is a relatively new area of study, relatively new compared to my career, which is long. As you stated, my lab was named Cell and Molecular Biomechanics. It does not include mechanobiology, because when I started out, the field of mechanobiology doesn't exist. So I like to maybe explain in contrast to biomechanics. It sounds like similar, maybe semantic, but there was big distinction, at least to me. So you had two words that are, combined to make a new words, right? Bio or biology, and then mechanics or mechano. Just flip the order. And so what's the difference? The way I understand it, and I explain it to my student, is that if you study a system, you have input to the system, and then you have output to the system, right? And here, both biomechanics and mechanobiology, the system's biological system. And then the input also mechanical input, like forces, stiffness of the environment, flow, pressure, that kind of thing. And then, but the output are very different. Biomechanics also look at the mechanical variable as output. For example, you apply force to bone, and how would bone deform? Whereas mechanobiology, the output is biological. So you apply force to bone, and how would the bone adapt the stresses to grow or resolve? So that's a big distinction. And in my lab, mostly we study at a much smaller level, so not bone as a tissue, but cell and molecules expressed on the cell surface.
Yeah, that's great. So speaking about your lab, you mentioned that you had a lot of projects that you're working on. Do you want to tell us about maybe just a few of them that are interesting?
Yeah, so in terms of the number of people in the lab that are working on the different project, the current lab, the biggest component is immunology, but there are different immunologically related things that we study. ranging from basic immunology questions like molecules to study. For example, antigen receptors on the surface of T lymphocytes or B lymphocytes, how do they recognize antigen, either the pathogen that coming outside of the body as intruder, or arrive from cells like cancer, or that cause autoimmune diseases. So those are T cell receptor and B cell receptor, and because they are important to health and disease, we study not only the basic biology or basic immunology of that with the using the mechanobiology lens to look at this problem, but there are important applications. For example, cancer immunotherapy, like infectious disease, viral infection. So we have, I think, 3 of my 4 current NIH U1, R1 grants are on immunology, and 2 are on cancer immunotherapy, infectious diseases.
Yeah, that's really interesting. So because we're talking about cancer and immunology, how do you work with how does your work related specifically to human health and cancer and disease help drive medicine and healthcare in those directions and like for treatments?
So we of course hope to contribute to those areas, right? But certainly there was a big biotech industry doing things like that as well. So I just use one project for example. So cancer, the most exciting recent development is immunotherapy, and there were 2 broad classes of cancer immunotherapy. One is immunocheckpoint blockade, and we have actually a project that funded by NIH, R1, working on that with the molecule called PD1, program that one. And then the other broad category is adaptive cell therapy. So the immune checkpoint blockade would be using antibodies to block molecules on cell surface to allow the cells that are being inhibited now getting activated. The other is using cell itself as a drug. You can either take patient's cells, or you can take a healthy donor cell, particular cell lymphocytes that they have the function, if they work properly, to attack and kill cancer cells. And the important thing is for those lymphocytes called T lymphocytes, T cells, to recognize tumor cell, and otherwise if you indiscriminately attack, then you kill normal tissue, so you get this side effect. So and the way that these T lymphocytes recognize the cancer through antigen is either through the native T cell receptor, or you put in a receptor that is man-made called the chimera antigen receptor, or CARs. So these are the 2 types. We work more on the T cell receptor, but we also engineer the T cell receptor. There were some pros and cons of the 2-type CAR T versus native T approach. I don't want to get into too much of the detail, but we are working on, based on our understanding of the T cell receptor signaling. 10 years ago, my lab published the first paper on cell showing that force amplify the antigen recognition of the T cell receptor and elicit stronger responses of T cell. So based on that understanding, we are working on engineering T-cell receptors for more efficacious T-cell receptor for recognizing tumor antigen.
That's great. Well, congratulations on the paper.
That's
So
10 years ago, so
yeah.
months also.
Mhm. So we're nearing the end here, and the podcast really tries to give students an understanding of research. So what advice would you try to give to students who are thinking about joining either your lab or looking to get into research in general?
So I think for students interested in, I guess, biomedical engineering, the biomedical engineering field is very broad. There are many things you can do. And students program have high credentials. Our admissions committee has done a great job in selecting great students. But you cannot do everything. So I think the success in this field really the most important factor, I think, is the interest and motivation of the student. So identify a particular project or lab that interests you most. Even when you join the lab, like my lab, it was different project. Like we work on immunology, we work on cancer, we work on cardiovascular diseases, platelets, and neurocells. You can still find the particular project that interests you most. So once you identify that, or even if you don't know in the beginning, try to get interested in it because you want to own the project. You don't want to just take instructions from the advisor, just execute it. Right? So that's what I think is important for a grad student to be successful in their graduate education.
Do you have any advice for undergraduates who might be looking as well?
I think it's the same otherwise, right? I thought your earlier question is for new students are selecting that, right? For
Yeah.
undergraduate, for undergraduate, if they are freshmen, they just explore the field. It's when I started my career, I was in mechanical engineering, so there was not a BME department, not many in the country, right? And there was a doubt that whether or not industry will hire a biomedical engineer undergrad or graduates from the bachelor degree. That's gone now. So the industry fully embrace the BME bachelor degree. But you need to explore. There are different engineering disciplines. There are also science disciplines. So whether or not you like it, you'll find out.
Of course. That's great advice. Thank you. So we are nearing the end of our time. So I'm going to end off with this last question. Where can listeners find you if they have any more questions about you, your lab, your research, things like that?
So, my lab website is listed in the BME department website, I think next to my faculty page. So if you go to find the Coder Department of Biomedical Engineering at Georgia Tech Emory, look for faculty website. And my name is the last because my last name Z started with Z. So then my faculty website has a link to all the information, phone number, email. And if you come to visit in person, my office is in the Whitaker building, 1210. My lab is next to my office. So.
All right, that's great. Thank you so much again for taking your time out of your busy schedule to come talk with us.
Well, thank you for the opportunity.
For sure. Thank you.